Other day, I was discussing with you the maturation of B lymphocyte, which is antigen independent phase, and then that leads to the development of B lymphocyte, which is in fact antigen dependent phase. And when we said antigen dependent phase, I said that it is two different types. One is TI antigen, that is supposed to be thymus independent antigen, and thymus dependent antigen. And I had mentioned that the thymus independent antigen mode of activation of B lymphocyte will be short lived and does not give rise to immune memory. At the same time, the thymus independent activation of B lymphocyte plays a major role in immediately attacking the antigen and trying to conquer the antigen through production of IgM. Only one type of antibody it is going to be produced. So it will not be polyclonal activation. In the sense, 5 different types of antibodies are not going to be produced. It will produce only one type of antibody, and that will be IgM. And we will come across a term later when we are going to talk about further activation of B lymphocytes through TD antigen, that is thymus dependent antigen, or T cell dependent antigen. A terminology called as class switching. Each immunoglobulin is grouped under one class, and there are 5 classes: IgG, IgM, IgD, IgA, and IgE. Okay. Now, if all these 5 classes of antibodies have to be produced, it will be only when the B lymphocyte is going to be activated dependent on T lymphocyte, that is T dependent activation. And in that case, first antibody anyway is going to be produced will be IgM. Later, it will switch over, that is called as class switching. Class switching. It will switch over to produce IgG, which will be the largest amount of antibody which will be in circulation in our system until the antigen disappears from our body. Okay. So in the diagram here, the numbers have been put 1 and 2. Okay. So in the case of TI antigen, the 1 refers to the signal transduction brought about by the cross-linking of the 2 IgM molecules by the antigen. And signal 2, that is number 2 is signal 2 brought about by actually a cytokine. You know, when we had dealt with the maturation process of the B lymphocyte, there is already IL-7 cytokine secreted by the stromal cells. Remember, the stromal cells are secreting IL-7. And that IL-7 will be there in circulation still, and that during the thymus independent activation of the B lymphocyte, it will come to the rescue of the B lymphocyte. When it is getting activated with an antigen, which is thymus independent or T cell independent antigen, at that time that IL-7 comes and binds to that receptor and gives second signal. And that signal 2 is more powerful than the signal produced by IgM molecule, signal 1. So signal 2 is essential for that B lymphocyte to undergo proliferation, that is multiplication by mitosis, and produce clones of B lymphocytes of the same type, recognizing the same T cell independent antigen, and produce copious amount of IgM molecule and tackle that antigen. Okay, TI1 is lipopolysaccharides, TI2 is supposed to be glycoproteins, flagellins, and proteins. Okay, so the thymus independent antigen will have its own role to play, and it does play some amount of role in acquired immune response to produce only one class of antibody, IgM antibody, and it does not last long. It is short-lived. By then, other antigens that are there produced by the pathogen will be encountered by the B lymphocyte, okay, or it will be encountered by the antigen presenting cells, and they will be presenting the antigen sometimes to the B lymphocytes in the form of follicular dendritic cells, okay, that is in the lymph node where the B lymphocyte has arrived with the low affinity receptor on its surface. Low affinity receptor on its surface, it has arrived and has come in contact with that foreign antigen presented by the follicular dendritic cell, or it has tackled that antigen on its own, and that antigen has linked the MIgM molecule, that is membrane bound IgM receptor of the B lymphocyte, cross-linked. See, once again it is shown as signal 1. It's a very, very weak signal. Okay, and that signal will be sufficient to activate the B lymphocyte. And that signal will be responsible for the signal transduction and switching on certain receptor genes in the B lymphocyte. So thymus dependent antigen, which helps in activation of the B lymphocyte, will first produce signal 1, and that also helps in endocytosis of that antigen into the B lymphocyte and then process it, and B lymphocyte acts like a antigen presenting cell here, okay, antigen presenting cell here. But I have told you that B lymphocyte is not a professional antigen presenting cell, but given the occasion it will process the antigen. Now when does it process the antigen? When the antigen load is very low. If the antigen is very low, that is 1000 times lower in concentration, then very small quantity, nanomole, micromole level of the antigen is present. That time B lymphocyte by itself can process the antigen. But if the antigen is present in large quantities, that will be processed by the professional antigen presenting cells, that is macrophages and dendritic cells. In any case, both these cells, that is B lymphocyte as well as the antigen processing cells, will present the antigen after process because it is an exogenous antigen. And I had mentioned when I dealt with the MHC complex, whenever exogenous antigen is processed, that is going to be presented on class 2 MHC. And that is the class 2 MHC that is shown on the B lymphocyte, which is now acting as the antigen presenting cell. And that is what's shown in this diagram also. Here, the class 2 MHC is presenting the antigen after processing. Okay. Now, that B lymphocyte, which has processed the antigen, when it presents the antigen on class 2 MHC, it is going to be recognized by the T cell, T helper cell, with its receptor TCR, T cell receptor, which has an affinity for class 2 MHC, which goes and receives the signal from the class 2 MHC by forming a conjugate with that. And this is the first synapse that is going to be established. The conjugate of the B lymphocyte, which is activated with a foreign antigen with T helper cell, will be established through class 2 MHC presenting the antigen to TCR. Simultaneously, the TH cell has another receptor called as CD4 receptor, CD4. That is why it is called as CD4 T cell. And that CD4 receptor simultaneously should establish the contact with the beta 2 domain of the class 2 MHC. When the class 2 MHC is presenting the antigen, TCR will establish the contact with the antigen presenting cleft of the class 2 MHC. Simultaneously, CD4 establishes the contact with B2 domain of the class 2 MHC. When these two establish the contact, that will be the signal 1 for the TH cell to get activated. That is signal 1 for the TH cell to get activated. At the same time, the B lymphocyte, which in fact had got activated with signal 1, had switched on some genes for production of the receptor. First receptor that is going to be produced by the B lymphocyte will be CD40 receptor, CD40R. And that CD40 receptor produced by the B lymphocyte on its surface is going to establish the contact with the opposite ligand, that is CD40L, that is going to be produced by the TH cell. Why? Because TH cell is also now simultaneously activated because it has been presented with the foreign antigen. Now, the beauty of the system is the humoral immune response that is going to be started with the B lymphocyte is also activated. The cell-mediated immune response that is going to be started with the foreign antigen is also started. TH cells are also activated simultaneously with the B lymphocyte. So then the CD40 that has established the contact with CD40L will establish such so firmly that it will produce signal 2. That is what it was shown here, signal 2. And that signal 2 will make that B lymphocyte to produce now furthermore receptors on its surface to establish the firm contact with the TH cell. Now you see, the first contact was through class 2 MHC with TCR plus CD4. The second contact was with CD40R and CD40L. Then the third contact will take place through two more receptors, that is B7 from the B lymphocyte and CD28 from the TH cell. Okay, so these receptors also going to establish the contact. By then, because of the contact of these three different receptor contact, this synapse is very, very firm now. And this B cell, T cell conjugate is very firm. By then, the B lymphocyte has gone into from G0 stage to G1 stage. Okay. Now, these stages where first contact with the antigen, processing the antigen, and establishment of the CD40. Then, after the establishment of the CD40 through the class 2 MHC, the presentation of antigen to PCR. Until then, it is considered to be competence signal for the B lymphocyte. Competence signal. And that competence signal is preparing the B lymphocyte to undergo mitotic activity. See, after G1, next stage is S phase. So by then, it should have established a contact with the TH cell with all these receptors. So B7 receptor is not only the characteristic of B lymphocyte, it is also the characteristic of all the antigen presenting cells. Macrophages also will express B7. Dendritic cells also will express B7. Any antigen presenting cell should express B7, which is supposed to be actually B7.1. Okay, and B7.2 when it expresses, that will be the stop signal. That will come at the end. We will not talk about it now. Okay, B7.1 will establish contact with CD28. So three different receptor contact is called as B cell T cell conjugate, which is a synapse. By then, the T lymphocytes, that is TH cells, as well as B cells have gone into G1 phase. Now next what happens? See. By then, the B lymphocyte, because of the establishment of that synapse, will switch on the genes for expressing the receptors for the cytokines. And those cytokines are responsible for stimulating the B lymphocyte to undergo mitosis. One. That is the cytokines produced by the TH cell. Because the TH cell is now simultaneously activated, it will produce the cytokines. And those cytokines will be IL-2, IL-4, IL-5, IL-6. Interleukin 4, interleukin 2, interleukin 5, interleukin 6, of which interleukin 4 and interleukin 6 play a major role in B lymphocytic process. Okay, the B lymphocytic process. Interleukin 2 is an autocrine factor that is responsible for proliferation of the TH cell. The TH cell also should undergo clonal proliferation with respect to that antigen that was presented by the antigen presenting cell, and they should now become effector cells to become TH1 and TH2 cells and T memory cells. That process we'll discuss separately with respect to T lymphocyte maturation, development, activation, differentiation. For the time being, you keep in mind that TH cell activation is simultaneous when the B lymphocytes are presenting the antigen to the TH cells. And presenting antigen to T lymphocytes through B lymphocyte and that synapse is a must to produce T dependent B cell activation that will produce long-term memory for the B lymphocyte. Okay. The cytokines now received by the B lymphocyte would put those B lymphocytes into proliferation. This is proliferation. But in the examination, I want you to draw only this diagram when I ask you for B cell activation. And I do not want you to draw these series of diagrams. Do not waste your time in drawing the series of diagrams. For T independent antigen, I would expect you to draw this one diagram. Plus only this diagram to tell that it is T independent antigen type 2. I had told you that it will not cross-link entire IgM, it will cross-link two adjacent FAB sites. T independent type 2 antigen. Okay, only this diagram. Not this. TS cell should not be shown. And TI antigen, that is type 1, you have to draw this diagram. And TD antigen, either you draw this diagram or this final diagram, this one, which is shown on top here in this slide. This diagram will be more than sufficient. And label these receptors. Label these receptors as labeled here, as labeled here. They are not labeled here, but you have to label them like this. So that completes the process of B cell activation where the B lymphocyte now undergoes proliferation. When it is undergoing proliferation, it is because of the influence of the cytokines, okay, especially IL-4. Large number of antibody producing genes will be switched on. Okay, so there are about 300 heavy chain genes, 300 light chain genes, and we'll be talking about that in detail when we study the structure of the antibody. But each time the cell is dividing, undergoing proliferation, it will now recombine one particular variable region gene among the 300 variable region gene with one constant region gene. The constant region gene belongs to 5 different classes: IgM, IgD, IgA, IgE, okay, and IgG. So these 5 different classes of the constant region constitutes the heavy chain. So heavy chain gene of each class should be recombined with the variable region gene, okay, recombined with the variable region gene, and that process will be taking place rapidly in the proliferating lymphocytes, that is proliferating B cells, and that will be taking place because of the influence of IL-4. Okay, and at that process, each time it will be putting up one MIgM molecule. So now this MIgM molecule that is put up on the B cell here will be different from what MIgM molecule was there earlier. All right. Earlier, it had low affinity to that antigen. Now it has to put in a MIgM receptor which will show the best and highest affinity to the antigen. So in permutation and combination, the clones of B lymphocytes that are going to be produced try to put their best MIGM receptor on their surface, which will show high affinity interaction with the same antigen against which they have undergone activation. Initially, it will be low affinity. Now, high affinity. But it is not possible for all the B lymphocytes that are going to be clonally proliferating. The clonal proliferation, millions of B lymphocytes will produce in that activation process. Of them, some of them will fail. In that variable region, recombination with the constant region, some of the receptors that are going to be put in will be once again with low affinity or no affinity. In such case, those B lymphocytes are going to be eliminated, which has been shown in this diagram. Apoptosis, low affinity, low affinity. Okay, and this stage of B lymphocyte which undergoes proliferation is called as centroblast stage. Centroblast. I told you blast is supposed to be for proliferation. So low affinity receptors, those B lymphocytes which have got low affinity receptors will undergo apoptosis. Only those B lymphocytes which have put in high affinity receptors will survive. Now those B lymphocytes in the germinal center, which have undergone proliferation, will migrate now to the medulla region. Those having high affinity receptor now migrate to the medulla region. In the medulla region, those B lymphocytes now have to undergo differentiation to produce the plasma cells. Okay, plasma cells. Plasma cells are large B lymphocytes, but they do not undergo proliferation like B lymphocytes. They will shed off the MIGM receptor and IgD receptor as well as Ig alpha and Ig beta. Remember, I had shown you that receptor when I had dealt with the initial stage of B cell maturation. Ig alpha, Ig beta receptor. These two receptors are also shed off. MIGM receptor also shed off and MIGD receptor is shed off. And after shedding off those receptors, the cell has to undergo transformation and now become antibody producing cell. And that process is called as B cell differentiation. And where does this process occur? That process occurs in the medulla region of the lymph node, this region of the lymph node, where the antibody production. By then, the B lymphocytes which were activated, which had undergone proliferation, which had established the contact with T lymphocyte, have descended from the secondary germinal center into the paracortex area, and in the paracortex area they have developed high affinity interaction with the antigen, and now they are ready to become differentiated, and they will respond to now T lymphocytes by responding to the copious amount of the cytokines secreted by the T lymphocytes, and those cytokines will give rise to different types of antibody producing cells. Okay, so the centroblast, which is showing the high affinity, okay, will now undergo proliferation with high affinity receptors on its surface now, and those cells now will respond to different types of cytokines secreted by the Th cell, and that cytokine will be IL-2, IL-4, IL-5. In addition, IL-6 is not shown here, but it is very important for differentiation of the B lymphocyte. Therefore, it is called as B cell differentiation factor. IL-6 is called as B cell differentiation factor. Okay, so that B cell differentiation factor makes these the centroblasts, which have become now centrocytes, which will not divide further, but do have high affinity receptor on their surface, transform themselves or differentiate themselves into plasma cells. Now, what is the characteristic of plasma cells? Plasma cells will be 1,000 times larger than the B lymphocytes without receptor on their surface, and they will push the nucleus to one side of the cell. Therefore, the nucleus is eccentric, and they will put in large amount of rough endoplasmic reticulum in their cytoplasm, which will occupy more than 3/4 of the volume of the cytoplasm, because that is required now for the protein synthesis. Rough endoplasmic reticulum is meant for protein synthesis, and it is also responsible for the post-translation modification of the antibody. The antibody that is produced will be glycosylated in the endoplasmic reticulum and finally dispatched out of the plasma cells. Okay, so the other different cytokines secreted by the TH cells will make these centrocytes to differentiate differently. For example, those centrocytes which respond excessively to interferon gamma secreted by the TH cell, and it is supposed to be one of the important cytokine, they will produce by class switching. I had told you first the B lymphocyte will produce only IgM, only immunoglobulin M will be produced, and that will be there for about 5 days in circulation. And after that, it is taken over by the IgG. And that mechanism I mentioned to you is class switching. Once the class switching takes place, it can produce different classes of the antibodies, and IgG class of antibodies, which can be further subclassified into G1, G2, G3, G4. We'll talk about them later in the later lectures. What are their properties? And the IgG class is going to be produced if interferon gamma is going to be the sole cytokine to trigger that centrocyte to differentiate to plasma cell and produce the antibody. Okay, and if it is TGF beta, tissue growth factor beta, the centrocyte is going to respond in such a way that it will switch over the mechanism to produce IgA class of antibody and IgG2B. Here it was G2A, it is G2B. IgA class of antibodies, if it is required, and it is required in secretory fluids. Let us presume that the B lymphocyte, which has undergone activation in the intestinal surface, in the mucosal associated lymphoid tissue, and it has encountered the antigen there, the foreign antigen in the mucosal area, that is mucosal area of the intestine, then that B lymphocyte should not produce other antibodies other than IgA. So in that case, it should respond to one of the cytokine, that is TGF beta, and produce copious amount of IgA because it can survive in the mucosal lining. And I had mentioned that IgA cannot be digested by our enzymes. It has a protection. It cannot be digested by the proteolytic enzymes. It has a protection by the inhibitor protein associated with it, which we'll talk about that when we are going to talk about the IgA in detail. Okay, so that B lymphocyte, which has transformed into plasmablast cell, will respond to TGF beta to produce IgA. Okay, then comes the another B lymphocyte. If it responds to IL-4 in large amount, then that B lymphocyte is going to produce the antibody, and that antibody will be responsible for allergic reaction. It is IgE antibody. IgE antibody is for all sorts of allergies in the body, and that has got receptor on the mast cells. As soon as it is produced, it will go and bind to the mast cells and create havoc. So now you know the B cell responds differently to different types of antigens and different regions of the body. Okay, so IL-4 will be responsible for production of predominant amount of IgE and G1, of course, and G1 G2 or G3, G4 will be produced as a class switching mechanism takes place because it is the next immediate adjacent antibody to IgM and IgT. But it will subside, will be taken over by other antibodies depending on the region of the body. So IgE will be the antibody for allergic reaction. Then suppose it is a particulate antigen. Let's say it is the bacterial cell itself, particulate antigen, or it is a viral particle. Then the class switching mechanism will not take place. The centrocyte will respond to the cytokines like IL-2, IL-4 and IL-5, which it had responded earlier, and it has started producing IgM as a first antibody. It will continue to do the same after plasmablast cells are produced, which will continue to produce only IgM, and it will not allow that plasmablast cell to produce other antibodies. There will be no class switching. Class switching will not take place, it will produce IgM. Why IgM? I had told you that IgM is a pentamer. It has got 10 antigen binding sites, FAB sites. And because of the 10 antigen binding sites, it can agglutinate a particulate antigen much more efficiently than IgG. If IgG is there, then large number of IgG molecules will be required for agglutination reaction. Therefore, IgG is considered to be a poor agglutinating antibody, but a very good bacteriolytic antibody, as well as it is a very good antibody for opsonization, whereas IgM is considered to be an antibody for agglutination and complement mediated lysis. Because IgM molecule having pentamers, those pentamers are readily placed at a distance of 100 angstroms of each other, and I had mentioned that when the complement is going to be activated, the C1q complement, which is going to bind to the Fc receptor CH2 domain of the antibody, the antibody molecule should be adjacent to each other at 100 angstrom distance, and two molecules of antibody will be required for complement to be activated. So therefore, two molecules of IgG are required for the complement to be activated, whereas IgM, one molecule itself will be sufficient. And it is a strong complement activator, IgM, strong agglutinator. These are the two different properties of IgM. So the differentiation of the plasmablast cells differs with response to the response of centrocytes to the cytokines that are secreted by the TS cell. And it also depends on the type of antigen that has made its entry and the type of antigen that was tackled by the antigen presenting cells as well as the B lymphocyte, and that was presented to the TH cells. Okay, and all this process which is happening, that is activated centroblasts undergoing proliferation, then the formation of the centrocyte and the plasmablast cells will have taken place in a lymph node in the medulla area and the paracortex area. The plasmablast cells will be here, it is shown here in the medulla area, that are called as plasma cells. Once the plasmablast cells are produced, they will transform themselves into plasma cells and they will start now producing the antibodies. Okay, and that antibody is released through the efferent lymphatic vessels into the blood. It comes into the circulation. And I had mentioned that largest amount of antibody will be IgG because it can diffuse out of the blood vessels. It can reach any part of the body by diffusing out of the blood vessels. Therefore, 65% of the antibody that is produced in our body will be IgG class. If it is IgM class, as mentioned here, that IgM class will remain confined to the blood vessel only. It cannot escape from the blood vessel. The antibody produced against our blood group AB antibody is IgM class. Okay, so it cannot escape from the blood. So if the antibody has to reach the fetus, that is the developing child in the mother's womb, it has to be IgG class. And that IgG class can diffuse through the blood vessels into the placenta and then reach the fetus and provide immunity to the fetus. And I had told you about this in passive immunity, naturally acquired passive immunity. In that, I had given this example where IgG diffuses from the mother, from her acquired immune response into the fetus, and it will provide protection to the fetus which is developing in the mother's womb. Okay, so the differentiation process is very essential to produce the antibodies. Okay, and this will happen only with thymus or T cell dependent antigenic stimulation. Okay, T cell dependent antigenic stimulation, and it will result in the production of acquired immune memory. Okay, various different types of antibody production. Class switching is possible only with T cell dependent antigen stimulation of the B lymphocyte. Otherwise, with the TD, it will remain only for the production of IgM, but there will be no long-term memory. Plasmablast cells also going to be produced in the TD antigen, but they will not show class switching mechanism. They will produce only IgM class and they do not last long. Okay, immune memory will fail. So this diagram shows an overview of the development of the B lymphocyte. It started with the stem cell. That stem cell gives rise to lymphoid lineage, and that lymphoid lineage will differentiate into two lines, that is one into T lymphocytic, one into B lymphocytic. And I mentioned that the B lymphocytic one will have a receptor on its membrane at CD45 receptor that makes that B lymphocytic lineage remain in the bone marrow, and that becomes progenitor B cell. And the progenitor B cell still does not have any receptor on its surface. It will establish the contact with the stromal cell with the VLA receptor on its surface with the VCAM, and once it establishes the contact with the stromal cell, it will become the pre-B cell, and the pre-B cell will start putting in the receptor on its surface as the membrane-bound receptor MIGM, and that MIGM will have a low affinity to the antigen, which is of the foreign origin, and it may have high affinity for the self antigen. And when it has got high affinity for self antigen, it is going to go through the negative selection, and those B lymphocytes are going to be eliminated. And when it shows low affinity to the foreign antigen, it will undergo positive selection, and that positive selection results in that B lymphocyte being selected, and that B lymphocyte now readies itself to receive the foreign antigen and becomes high affinity antigen receptor on its surface, and then it will transform itself to become the plasma cell, that is differentiation, and that will happen in the peripheral lymphoid organ with the high affinity to foreign antigen. Then it will start producing the antibodies, and first antibody to be produced is always IgM, which is for 5 days, then it disappears, then it is taken over by other antibodies, that is IgG, IgA, IgE, because of the class switching mechanism. Okay, so the response of the B lymphocyte that is shown, you can graphically represent it like this. When the first antigen contact takes place, that is called as primary response. The response is very, very small. You see the concentration of IgG, see the concentration of IgM, and the lag phase. And this refers to 1 month period. Within 1 month, how much of the response has taken place? Only small quantity of antibody is going to be produced. But when there is a booster means second contact with the antigen, the response will be enormous, and the secondary response is supposed to be through the memory cells, and memory cells will respond very quickly. So, primary response when it takes place, the B lymphocytes take about 7 days to produce the antibody IgG type, 5 days to produce IgM type. So 14 days to produce good amount of antibody to tackle the antigen. But memory cells will take 24 hours. Those B lymphocytes which are come in contact with the antigen for the first time, when they come in contact with the antigen for the second time, they will get activated within 24 hours. They are kept in reserve in the bone marrow. Therefore, the secondary response will be always high with IgG, with high titer antibody, and IgM response will be lower than the first response. It is always first IgM molecule going to be produced, then taken over by IgG. Keep remembering that that mechanism is called as class switching mechanism. So I'll stop here. Thank you.